Ticks and tick-borne diseases are increasingly becoming a major health concern for humans, domesticated animals, and livestock. Disease cases from infected ticks, mosquitoes, and fleas have tripled in 13 years, and greater than 80% of these disease cases are transmitted by ticks. Ticks are highly efficient vectors of disease agents because of their abundance, their host preference, longevity, and ability to modify the site of attachment. In the United States, ticks transmit at least 14 bacterial pathogens responsible for 9 different diseases. Ticks also transmit at least 4 viruses and 1 protozoan agent responsible for babesiosis. The majority of tick-borne disease cases have been reported from the Northeast and upper Midwestern United States. There are 3 important tick species in Connecticut and throughout the United States. However, black-legged ticks, also known as deer ticks, constitute greater than 80% of ticks in Connecticut. The black-legged tick is widely distributed across the eastern United States and transmits pathogens for Lyme disease, anaplasmosis, tick relapsing fever, ehrlichiosis, babesiosis, and Powassan virus disease. During 1996 to 2015, the number of counties in the United States with established populations of black-legged ticks has doubled. Numbers and geographic distribution of Lyme disease cases mirror other tick-borne diseases, and as the human cases of Lyme disease has increased, so has the number of other tick-borne diseases. The life cycle of black-legged ticks generally lasts 2 years. During this time, they go through 4 life stages: egg, larva, nymph, and adult. After the eggs hatch, the ticks must have a blood meal at every stage to survive. Black-legged ticks can feed from mammals, birds, reptiles, and amphibians. The ticks need a new host at each stage of their life. The American dog tick is widely distributed east of the Rocky Mountains and also occurs in limited areas on the Pacific coast. This tick transmits pathogens responsible for tularemia and Rocky Mountain spotted fever. The Lone Star tick is widely distributed in the eastern United States, but more common in the south. This tick species transmits human ehrlichiosis, tularemia, Heartland virus disease, Bourbon virus disease, and southern tick-associated rash illness, or STARI. This tick could also cause alpha-gal syndrome, a recently identified type of food allergy to red meat. As an aggressive human biter with highly irritating bites, the Lone Star ticks have been established in the southeastern United States for well over a century. Southern New Jersey was historically recognized as their northern range limit. However, in recent years, the range of this tick species has expanded northward and reached Connecticut. These photos depict heavy infestation of a deer in Manresa Island near Norwalk, Connecticut. The number of Lone Star ticks submitted to the CAES tick testing laboratory increased by 58% from the time period of 1996 to 2006 to the time period of 2007 to 2017, mainly from Fairfield County. Established populations of Lone Star ticks were discovered in Fairfield and New Haven counties in 2018 and 2019 respectively, and further establishment in New Haven County was documented on June 17, 2020. The ongoing interception of exotic ticks parasitizing humans entering the U.S. are of major concern. Recently, in 2017, established populations of the invasive Asian longhorned tick were discovered in New Jersey, and it has now spread into at least 14 states, including Connecticut. This tick is native to the Korean Peninsula, Japan, and eastern regions of Russia and China, and it is a major livestock pest in Australia and New Zealand. It is a vector for the viral agent of severe fever with thrombocytopenia in humans, among other pathogens. Furthermore, the recent detection of the more pathogenic Theileria orientalis in cattle and in ticks in Virginia, for which Asian longhorn tick is a known vector, is suggestive of risks to livestock with the introduction of exotic tick species. The CAES Tick Testing Laboratory was established in 1990 following the discovery of a group of children with arthritic and other symptoms in Lyme, Connecticut. The program was mandated to test ticks for Lyme disease pathogen. However, in 2015, the program was expanded to include testing for the causative agents for babesiosis and anaplasmosis, respectively. Every year, greater than 3,000 ticks are submitted to the tick testing laboratory by residents, health departments, and physicians' offices. However, in recent years, the number of submissions has reached near 6,000. Ticks submitted to the CAES Tick Testing Laboratory are examined for species, life cycle stage, and engorgement status. Engorged nymph and adult female black-legged ticks are screened for evidence of infection with disease agents. The testing procedure involves DNA extraction and screening DNA by PCR using diagnostic genes. Tick testing results are reported to the residents, health departments, and physicians' offices by email. Of the 35,071 ticks submitted to the CAES Tick Testing Laboratory between 2010 and 2019, nearly 85% were black-legged, also known as deer tick, 12.4% American dog tick, 2.5% Lone Star tick, and a very small percent other tick species. The number of all three tick species, including black-legged, American dog, and Lone Star ticks submitted to the CAES Tick Testing Laboratory has increased during the last decade. Submissions of nymphal and adult female black-legged ticks follow a distinct seasonal pattern. Nymphal tick submissions peak in June, while submissions of adult female ticks show a bimodal pattern with a major peak in April through May and a minor peak in November. The June peak of nymphal submissions coincides with the June through July peak in reported Lyme disease cases in Connecticut. Greater number of ticks submitted to the CAES Tick Testing Laboratory are those biting younger children, followed by adults in their 50s through 60s. Over the last decade, a total of 35,234 ticks have been submitted to the CAES Tick Testing Laboratory, with the lowest number of 2,593 in 2010 and the highest of 5,577 in 2017. The average infection rate has been around 32%, but in recent years it has increased to 36 to 38%. In addition to increases in Lyme disease pathogen infection rates in black-legged ticks, infection rates for anaplasma and babesia have also increased in recent years. Similarly, the percentage of black-legged ticks simultaneously infected with 2 or 3 disease agents has also increased. At the town scale, we found evidence of spatial clustering for the rate of submitted black-legged ticks. The majority, 81% of submitted black-legged ticks, were from Fairfield and New Haven counties. However, prevalence of infection with Lyme disease pathogen in black-legged ticks was higher in more rural counties. We also note differences in Lyme disease incidence across counties in Connecticut. Lyme disease incidence was highest in Windham, Tolland, and New London counties, and lowest in New Haven, Fairfield, and Hartford counties. We also note high incidence towns at the intersection of Tolland, Windham, and New London counties, and low incidence towns in southwestern Hartford and northeastern New Haven counties. Tick-borne diseases will continue to be a major health concern. Additional pathogens will emerge, and rate of co-infections in ticks will likely increase. Range expansion of ticks and tick-borne diseases will continue. The Lone Star tick is an important emerging health threat in Connecticut and the Northeast because of increasing population densities and subsequent range expansion, its non-discriminating biting habits, and its capacity to transmit diverse pathogens. Curtailing the geographic expansion of these ticks and the spread of infection in humans and livestock is vital to prompt interception and identification of invasive ticks and pathogens, and to implement efficient eradication and preventive measures.